So why don't we go ahead and get started? You'll notice we've got these posters of butcher cuts up at the front. If you have a hankering for knowing exactly what cut of meat covers your information, you know, where your flank steak or your ribeye or your loin comes from, then you can take a look at those. Let me also stop right when we get started and let me acknowledge some special guests that we've got with us. So we are really honored to have Dr. Carl Huether and his wife Carol that are with us today. Dr. Huether actually has been a huge advocate of human genetics education. He started the genetic counseling program at the University of Cincinnati back in the 80s. And a number of graduate students have worked with him on the focus of human genetics education. Dr. Adam Hott, who's a member of my team, came out of Carl's lab, and Dr. Bethany Bowling, who's with us as well, came out of his lab. Bethany's spending a month with us, helping us put some education pieces together. So he has played a major role in shaping how students know about human genetics, and he's here at HudsonAlpha for a couple days. And Dr. Huether, thank you very much. We're really glad to have you. So we talked last week when we talked about crops. We talked about some of the reasons why seed breeders and farmers look to enhance their crops, look to specifically make selections. And what they're doing when they're making selections, when they're looking for specific traits, is they're choosing alleles, genetic combinations, genetic flavors that provide the traits that they're interested in. We're going to see that exact same thing borne out this week as we talk about livestock genetics as well. So the goals of animal improvement: lower inputs, less food, primarily less food, but higher outputs. Same sort of thing that we saw when we were talking about crops. And then increases in specific traits that are really desirable. Yield. Can I get more meat? Can I get more wool? Can I get more of what I need? More milk, more eggs. Life cycle characteristics. Is it easy or easier for the animals to give birth? Are they fertile for a longer period of time? How rapid do the individuals grow from birth to the first couple of years or the first few months before they're ready to go to market? What's the flavor? What's the marbling, the tenderness, those sorts of pieces? How hardy are they? This is, you know, we don't necessarily think about this, but if I am breeding and growing cattle in Minnesota, will those same cattle do equally well along the Rio Grande? Very, very different conditions. If I'm growing chickens in a hen house and I'm doing that in California, how well does that translate to a hen house in Virginia? So do they grow under multiple different kinds of conditions? And then aesthetic value. And I think one of the pieces for aesthetic value, something that we find attractive, I think it's an excellent night for us to actually talk about that specific piece, because you're all here instead of at home glued to the television, because tonight is the finals of the Westminster Kennel Club. And if that is not aesthetic value for dogs, I don't know what is. So these are some pictures from yesterday. And you know dogs, those of you that have dogs, those of you that have seen dogs, dogs have an enormous amount of variation. Just the differences from breed to breed in size and hair color, in the length of the hair, in the shape of the face, the shape of the tail, enormous amounts of variation. This is a great poster, those of you that are dog lovers, from Pop Chart Lab, a diagram of dogs, and you can zoom in and you can see it covers lots of different breeds. So here are the hounds, and here are the toy breeds, and it is amazing the amount of variation that we have from dogs. And it's even more amazing because dogs were domesticated somewhere in the 25 to 15,000 year ago time frame. We've argued a lot about this. We thought dogs were domesticated 30,000 years ago, and then a paper came out earlier this week that said that the skulls that we thought were dog skulls are really wolf skulls, not dog skulls, so maybe dogs were domesticated a little bit later. But anyway, dogs were domesticated from wolves that likely had some genetic alleles, some genetic changes in brains that made them less afraid of humans. These wolves were more likely to hang out around civilizations. And over time, they discovered that there was food near civilizations. And so we slowly domesticated them, but then from a relatively small number of dogs, we started these breeding programs to end up with dogs that are great hunters, dogs that are great herders, dogs that are lap dogs. So the amount of variation visibly between the breed of dogs is amazing. What's even more amazing to me is that some genetic research that was done probably about 3 or 4 years ago suggests that most of the variation that we see in dogs, ear shape, hair length, hair color, body shape, snout size, really is controlled by probably about 50 genetic spots. 50. Let me put that in context for you. We think that there are over 200 genes that control human height. 200 that control human height. And I just told you that there's probably 50 or so that contribute to all the physical differences that we see in dogs. It's an amazingly small amount of variation. But we also see variation in lots of other important agricultural animals like cattle and poultry and sheep. And again, the vast majority of this variation is due to changes in our genes. So these alleles, this genetic variation, that adjusts the recipe in very small ways, but contributes to all the variation that we see. We're going to spend most of our time talking about cattle and chicken, but I wanted to just also note the amount of variation that's present in pigs. This is a poster of different breeds of pigs from Latin America. Just the shape, the coloring, the size of the snout or the smashed-in face. My favorite pig is a Mangalitsa pig. Now this pig, yes, has curly blonde hair. Long, curly blonde hair. These pigs are primarily found or have been bred in Hungary and parts of Czechoslovakia. They were quite popular before the Communist Revolution. It looks like a sheep. And this next part, I am not making this up. If you go back and you look through Jim Henson's original notes, Miss Piggy is a Mandalitsa pig. So that is the specific variety that she is modeled after, a Mangalitsa pig. There are some clear genetic differences in Miss Piggy compared to the rest of her breed, but I got a big kick out of the fact that she actually does have, I mean, they scientifically went through and figured out exactly what she looks like, you know, what breed of pig she would be. And we will leave the pigs alone at that point. We'll stop there. All right, if you remember on our first week, I gave you several important points, talking points that we needed to remember throughout the course of this series. And one of them used the balance beam behind me, recognizing that when we talk about agriculture, we are always going to have to balance out competing interests. There are groups that are going to argue that this is most important, and there are groups that are going to argue that the consequence of that has big problems for this over here. Let me give you an example of one of these balance points, specifically as it relates to livestock. And this is worldwide meat consumption. Now, this may be a little fuzzy on your handout, so I apologize for that. But what I want you to see, these data come from the FAO out of the United Nations, and they are looking at meat consumption per person in kilograms. And on the left and on the right are the same world map, but we're showing you different countries, their meat consumption. So Canada, the United States, the European Union, Australia, New Zealand, and Japan on the left, and on the right, the BRICS nation, Brazil, Russia, India, China, and South Africa. And the different colors correspond to different types of meat. So the purplish on the left is beef, the pink is pork, the yellow-orange is chicken, and the burgundy, which is on the far right for each of these graphs, is lamb. You'll see that there are two different bar graphs for each color. The lighter color is consumption from 2010 to 2012, and then the darker color is estimated consumption in 2022. So you can see kind of current consumption and what we estimate. There are a number of things that you could glean from this map. We could spend a lot of time on this. Let me just hit a few of them. We're looking at developed or developing nations here. If we were looking at underdeveloped nations, we would see very little meat consumption at all on any of these graphs. So recognize that we are looking at primarily first and some second world nations that are quite accustomed to eating meat, not only every day, but multiple times a day. That is a relatively recent trait in the history of the world. And for most of the world, meat is not something that happens every day. And as nations begin to become more developed and more affluent, eating meat every day or multiple times a day is something that they aspire to. So there will be more and more meat consumption as the countries become more and more affluent. But what I want you to see, let's look at America right here. So again, this is beef, pork, chicken, and lamb. Not a whole lot of lamb being eaten in the United States. A fair amount of beef, although beef is projected to actually drop in consumption by 2022, and I'll show you a map of that in just a second. Chicken, way up there. This is in kilograms. It's equivalent to about 48 pounds of beef and about 100 pounds of chicken per person a year, just to give you a frame of reference for that. You'll also notice that in India, which is a primarily vegetarian nation, not a whole lot of meat consumption at all. You'll notice lots of pork in the European Union and in China. That's the predominant. But in other places, chicken seems to be the predominant meat consumed. And then if we look at the United States specifically, and we dig into that a little bit more, you can see from the early 1900s through 2012 what the per person consumption of turkey, which bumped up a little bit in the late 80s, chicken, which dramatically increased from about 1950, and in about 2010 overtook beef as the most popular meat consumed in America. Pork, which goes up and down but has stayed pretty steady overall. And then beef, which hit its high point in the mid to late 70s and has been on a slowly downward trend, in part because Americans have eaten less beef because we've become more health conscious and have chosen to eat chicken instead. Now, whether that's an appropriate decision or not is not what we're going to talk about today, but you can see pretty aggressive marketing campaigns as Americans have chosen to eat less beef and eat more chicken. So that rising consumption, rising worldwide consumption, as more and more people around the world become economically more advantaged, they eat more beef, they eat more chicken, how are we going to balance out more livestock being farmed and produced against the following? There's a huge competition for resources. You need land if you're going to farm herds of cattle. You need water. You need feed. So you've got to have lots of, whether it's corn or whether it's grass or other kinds of grain, how are you going to grow the food? And there's a huge competition right now. It's called the food, feed, fuel competition. So let's just look at corn. Corn is used for human food, corn is used for animal feed, and corn is used to make ethanol for biofuels. So you've only got so much corn that you can plant, and you've got this competition across these three different things. So who makes the decisions about where our resources are, where they go, how we distribute those? Livestock created pollution. Livestock creates a lot of waste. And while manure in general can be used as a fertilizer, it also releases large amounts of ammonia. And animals release a fair amount of greenhouse gases. And if we're going to dramatically increase the number of animals that we're going to have on the planet that we are created for livestock purposes, we're going to dramatically increase the amount of waste that we need to take care of and the amount of greenhouse gas that's emitted. So how do we balance that out against denying people the right to have beef or denying people the right to have chicken? Animal welfare. This is a big one, and I'm going to note that it's here, but we're not going to dig into this. That's beyond the purpose of what I want to talk about. But how do you humanely raise large amounts of animals? And by the way, just because something is marketed as free range That means it has the opportunity to go outside, not that it is forced to go outside and walked around. So be aware of that. It's free range because it has the opportunity to go out. But if it chooses to stay inside by the food feeder, it's still marketed as free range. So again, remember, one of our key objectives for these 4 weeks is to recognize that what we sometimes think is natural or a certain way may not necessarily be that way. I'm not telling you that you need to change your views, but I want you to have your view from an informed perspective. So my job is to bring you the information, and then you make the decisions based on the information that's in front of you. And then robustness under different conditions. If all of the animals that we are breeding for commercial growth have been designed to grow in the southern part of the United States, that is not going to help us if we're going to send those same animals to Siberia. So how do we make sure that we breed animals that will grow well under commercial growth conditions in lots of different places so that all parts of the world have access? All right, so this is the balance point. We like meat. Lots of the world is liking meat even more. Are we going to deny people's options because of these pieces, or are we going to say these aren't important, let's eat beef, or let's eat chicken, or let's eat pork? So how do we balance those out? And again, I showed you this last week. This was talking about how we traditionally breed crops and how we take an elite crop that has all the traits we want except maybe for one. So this tomato that was here on the left is susceptible to pests. And then I take this donor plant over here that is resistant to pests but doesn't have any of the other qualities that I want. So how do I design a breeding program that lets me breed in the resistance to pests but make sure that I don't lose any of the elite varieties that I want from this plant? And so we talked about this concept of back crossing, so I am continually breeding generation after generation back to my elite plant, and I am reducing the amount of genetic information contributed by my donor only to the place that I am selecting, only in this instance to the fact that it's resistant to the pests. This exact same storyline takes place when we're talking about animals. So here's a sample livestock breeding between a bull from breed A and a cow from breed B. And I've just colored the chromosomes differently just to indicate different alleles, different information there. And so if this is their offspring, 50% of the genetic information from A and 50% of the genetic information from B. And if I back cross her back to the same type of bull, a bull from the same breed A, now I've got 75% of A, the black information, and 25% of B. And if I continue back crossing, I get less and less of this animal. But there may be a specific thing I'm selecting for. Maybe this animal has great milk quality. So I am only selecting out of all the births from this process, I'm only selecting the cows that have the milk quality that I want. So I'm specifically selecting for that specific little white bit of genetic information, those key alleles that give me that new trait that I want to add in, but I'm trying to keep all of the other traits from the black. So the exact same process. May take a little bit longer because generation times may be longer for animals. May take a little bit less time. If we're talking about chickens, that breeding window is relatively small. It's certainly smaller than a growing season. So I can get more generations of chickens than I can generations of corn. So, but the exact same concept. So let me stop and let me see if we've got questions at this point. It's a good place for us to take a break and survey the audience.
Why don't you list fish on your meat?
Why don't I list fish? I could have listed fish. Fish is definitely a protein source. And the same sorts of things happen with fish, especially those fish that are farm-raised. It's a little bit more difficult if we're out in the ocean to try to force breed to happen. But yes, fish, we can do the same thing. In fact, next week when we talk about genetic modification, we're going to talk about a variety of genetically modified salmon and some of the challenges that that faces in coming to market. So excellent point. Other questions? Let me see if we have any online. Not yet. Anybody?
What happens to the cows that don't have the good milk? The ones that are like, they're not in this progression.
What happens to the cows that don't make good milk? Those cows find their way to the feedlot and ultimately to the slaughterhouse. So that's a really good point, Gary, is that if I'm a herder and I have many head of cattle, I've got two different groups of cattle that I'm paying attention to. I've got the cattle that I'm going to send to the feedlot and sell for meat, and I've got the cattle that are forming the next generation, that I'm breeding to create the next year's population or a couple years down the road. So I'm paying attention, as I'm looking at my herd, I'm paying attention to which one of those bulls and which one of those cows have the qualities that I want. And then I may make them part of my breeding stock. But they may not have the qualities that I want, and so off they go to the feedlot and the slaughterhouse. And we'll jump into that in just a second.
So where do genomics come into play at this point? Are you just physically looking at the cow?
Yeah, well, no, it's more than just physically looking at the cow. We're going to talk about something, I think, in maybe 2 slides that'll answer that question, and genomics will probably be 3 slides after that. Was there, Bob?
Yeah, two related questions. Is dog a well-defined species or category of animals? And is it the most varied in phenotype?
So is dog a well-defined species, and is it the most varied in terms of its phenotype? Dog is a well-defined species, yes. Within that species, it has breeds. Those breeds can cross with each other. I am now going to stand right at the edge of my knowledge because traditionally, one of the definitions of species was that it could not breed with something else. It had become divergent enough, it had become genetically different enough that it couldn't breed back with whatever it originated from. I'm not 100% certain if that's the case. I think that you can breed certain types of wolves with dogs. But even still, there's enough genetic difference that under a slightly different classification scheme, we would call them their own species. Now, your second question, are they the most varied? I don't know, because I could think about a certain kind of flower. And there is a huge amount of variation maybe within that set of flowers. If you think about mums, how much difference there are not only in color but in size and all those pieces. So I'm not 100% certain that we would say that dogs are the most varied. Dogs show the most, that's not even true. I was going to say dogs show the most evidence of human intervention, but again, flowers would as well. But dogs are the ones that we have clearly put a lot of energy into creating these unique species. Good question.
Your diagram on worldwide meat consumption shows a 10-year block from first to second. Is that with a steady state human population or the anticipated growth of human population over the 10-year period?
That's a great question. So the worldwide meat consumption image that I showed shows the 2010, 2012, and then 2022 estimation. And my understanding is that that was based on a rising population as well. It wasn't based on if we had the same number of people that were still there. It was based on whatever population trends for those countries show. So for example, we would see a different population trend maybe for India than we might expect to see for the European Union. I had a question from online. Would I please define what I mean by back crossing? I'd be happy to. So back crossing is when you actually, you cross 2 animals. So in this case, breed A and breed B of bull. And then I get a hybrid that contains genetic information from each of the 2 breeds. I'm back crossing when I then mate that hybrid to an animal that is identical or very similar to one of the 2 parents. So in this case, I am mating this animal to the same type of bull as we had here, as her father was. And then, again, this cross produces this cow, and I am back crossing again, right back to the exact same kind of breed. So multiple rounds of back crossing increases the genetic information from whatever it is I'm back crossing with. So I am minimizing the amount of genetic information that came in from the other original parent. Good question. Any others?
How do you ensure the purity of the back cross? How do you know that that is exactly the same?
Yeah, how do I ensure the purity of the back cross? So the back cross bull is a variety that I have consistently bred from within the same breed. I've only made sure that I'm crossing it to other bulls or cows that have not outbred to anything else. It's much easier now that we've got genetics, because I can use genetic information to know, is this purebred or not? It was much more difficult when you were just going solely on looks alone. Others?
On crossbreeding, there's horses and donkeys, so you're cross-species breeding. Do you project that that's going to continue as knowledge advances?
Yeah, so cross-species breeding, like horses, I'm going to get this completely wrong. Horses and donkeys give you mules, is that right? Okay. And generally, but not always, those crossbreeds are sterile. Now, that's not always the case, and in part, that's because the genetic information, in order to make eggs or sperm, the genetic information from the parents, so let's say we're going to make an egg in this cow, the genetic information that she got from her mom and dad have to be compatible enough, because there's a process called meiosis that happens where chromosomes line up and divide. And if they're not similar enough, then those precursor egg or sperm abort. They don't form. So you end up with sterility. I think that we will, I'm sorry, and your question was, do I think we will continue to see crossbreeding? Cross-species breeding. I think probably we will, especially in those rare instances where it actually is fertile, because then there may be some really interesting characteristics that are chosen or selected in the population. Excellent questions, guys. Really good. Let me see if we have any more from online, and then I'll move us through. Does traditional crossbreeding include actual DNA analysis to verify the desired markers? Historically, crossbreeding didn't require any kind of genetic analysis. Today, the genetic analysis may be available for a species, and so we may use it, but you don't have to have genetic analysis in order to go through traditional crosses. So the things that Gregor Mendel did 100 and something years ago in his pea gardens, that you may remember from your biology class, that was crossbreeding. Didn't have any genetics at all, no genetic markers. Let me back up. He certainly had genetics. That was the underpinning of it. He didn't have any genetic markers that he could. That's not true either, because he saw the visible traits of the genetic markers. He didn't have any molecular DNA testing. Let's just go with that. All right, let's move on. So let's spend some time talking about cattle. So cattle were domesticated probably starting somewhere around 10,000 years ago from this now extinct animal called an auroch. And so this is a representation of an auroch. The last auroch went extinct in the 1600s in Poland. But they used to be wild animals that roamed all over the world, and cows were domesticated from the auroch. So there were probably multiple domestication events. One of them happened in the Fertile Crescent in Mesopotamia at about the same time that we were also domesticating barley and grains. And that was about 10,000 years ago, and that's called the humpless cattle. And that's the cattle that when you see like a Longhorn or a Jersey, that is the example of that domestication. But then a few thousand years later, there was another domestication in what is now modern-day Pakistan, and that gave rise to the zebu. Now this is the zebu. Let me move out of the way so you can see the zebu. The zebu also was domesticated from the auroch, but it has this big hump right here. So it's called the humped cattle, and it has this really saggy space. Let's just go with that. But it is also a domesticated cow. And then maybe, we're not 100% certain, there might have been a third domestication event in Africa, but we're not 100% certain about that. Scientists still argue about it. But we've got at least 2 and maybe 3 different original domestications, which means depending on what specific animals were chosen for the domestication, we're going to have some different genetic variation. Obviously, you can see because we don't have a hump in this type of cattle. We've got some differences to work with. So then from there, we now have produced over 800 different breeds of cattle. 800. Milk cattle, beef cattle. We've kind of split those among 2 different groups. We've selected the dairy cattle on the basis of the quality of the milk that they give, and we've selected beef cattle on the basis of the quality of their beef. Now clearly, you could get meat from a dairy cow, and you can get milk from a beef cow, but they've been selected because one is better at one and one is better at the other. Those of you that grew up on farms, you probably didn't necessarily have it. This was our beef cattle and this was our milk cow. We had a cow and it gave milk, and when that gave out, we had steak. That sort of piece. But let me give you a few examples. So the Texas Longhorn, this breed of cattle was descended from the original cattle Christopher Columbus brought over to America. And it was bred with some additional cows that were brought over in subsequent trips, ultimately crossed to the mainland, and thrives really well in Texas. And it's characterized by this really impressive horn spread. Now, the Brahman, these cattle were developed in America, but they were developed from 4 of the Zebu, 4 of the breeds of cattle from India. And they are incredibly heat tolerant and they're disease resistant. So we've got the hump that you recognize from the Zebu. Doesn't look quite like the Zebu, so because of some of the different breeding. We have the Angus, which is the most popular beef cattle in America, thanks to a really impressive advertising campaign, Certified Angus Beef. It was actually developed in Scotland. And when it was first brought to America, it was considered an oddity, and it wasn't actually mated. It was just looked at. But it's now the most popular beef in America. And then we have the Brangus, which is a little bit like what happens when you take 2 movie stars and they get married and you just combine their names like Brangelica. So we have the Brangus. And the Brangus is 3/8 Brahman and 5/8 Angus. And so it combines the heat tolerance of the Brahman and the meat quality of the Angus. And you can see, if you look at it, we got a little bit of a hump here, but not as much as we have in the Brahman and not nearly as much as we have in the Zebu. So you're seeing that some of these, as we crossbreed these, we end up with differences in the physical, the phenotype, the traits that we can see. And again, I'm not showing you any dairy cattle, the Jersey and the Holstein and all those. I'm not giving you any examples of those, but recognize 800 different breeds of cattle. Now, Gary, this comes back to your question. How did we say, this is the kind of cattle that I want? Did we just look at it and we said, okay, that looks kind of like what I'd be interested in? Here are some of the traits that cattle breeders and farmers are interested in as they build a herd. This is probably a little bit easier to see on your handout, but calving ease. So for example, are the calves big or small? How easy is it for those calves to be born? And then right underneath it, calving ease maternal, what's the hip structure, the bone, the pelvic structure of the mother cow look like? Is it easy for her to give birth or not? So the whole process of giving birth has to do with how well the pelvis is shaped in the birth canal and how big the calf is. So there are traits that you look for. We know that they're genetic, but historically, you just look to see, well, that looks like that was pretty easy for this cow to give birth. And it looks like that when I mate this bull with this cow, the calves are generally small, so it's easy for it to give birth. Birth weight, that's pretty straightforward. Weaning weight, how much does it weigh when it shifts from drinking mom's milk to a grain diet? Yearling weight, when it's a year old. Milk, how much milk does the mother give? If I now look at the carcass itself, once it's gone to the slaughterhouse, how much marbling is there? How much fat is there in the meat? What's the size of the ribeye? What's the weight of the carcass? How much food does it take in? How tender is the beef? How tall is the animal at a year? All these different pieces. So these are traits that if you're going to build up a herd of cattle, you want to pay attention to all these. Now, you can't maximize every single one of these. Obviously, if I want bigger animals, it's going to be really hard for me to have really big animals when they're time to go to the slaughterhouse and have really small babies being born. Those don't work that well. I'm selecting against each other. So you're going to have to make some decisions if you're the farmer when you're trying to decide what you're going to choose to be the parents of your next generation of your herd. So we started out with just looking at these, and then we created a set of data called Estimated Progeny Differences, EPDs. We're going to spend a lot of time talking about EPDs, so we're going to get comfortable with EPDs. That is saying, okay, for this breed, what's the average birth weight for this breed? Okay, let's say that it's 100 pounds. Then I'm going to say for this specific bull, and EPDs are often done for bulls, sometimes they're done for the cows, but not more so now, but historically they were done for the bulls. I'm going to say for this bull, when this bull impregnates a cow, how much do his calves weigh? So I am looking to see how much more or less his calves weigh than the average. So the EPD is a measure for a specific animal about how much a set of traits are going to differ from the population average. So in other words, it's kind of saying, okay, this bull tends to produce really large calves, and this bull's daughters tend to produce lots of milk, and this bull's sons and daughters have lots of marbling. So they are better than the average for the population. So they would have a positive EPD for those traits. This bull over here produces really small calves that don't have a lot of marbling, and this bull's daughters don't produce a lot of milk. So they would be less than the population average for that breed, and their EPD numbers would be negative. Let me give you some examples, because this is kind of a tricky topic, a wonky topic to wrap our head around. EPDs are built on a couple of different things. The pedigree of the animal, so where it came from, its progeny, and its own measurements. So here's the bull that we're going to measure EPDs on. And we're going to look at its parents and grandparents, and we're going to look at its children. So that kind of information, I know at birth where its parents and grandparents are, and I know how they performed. So that's, that concept of past performance is, you see on the stock market stuff, past performance is no indication of future earnings. In this case, past performance is a big indication of future earnings because I know the family that it came from. It's got good breeding. So that's going to inform the EPD measurements. But then I also have to wait until this bull sires enough children that I can gather the measurements on that about those children and then filter that back in. So I may not have good EPD measurements on this bull for several years. I'm looking at what its family is, and I'm looking at the children that it's produced. And then once it's produced several kids, once it's got 20 children, I can then say, based on its children, its next set of children are also likely to have these kinds of traits. And that's really important if you're trying to decide if this is the bull that I'm going to choose to build my herd, and it's especially important if you want to sell this bull's semen on the open market at $50 or $100 or $400. Because you need to be able to show that this bull really is going to be able to produce the things that you need, that it really is a stud muffin. That is an excellent point. Yes, EPDs are only based within the same breed. So I cannot compare a Longhorn to a Brangus. They have, because their population average is very different. So it only works if I'm comparing them within the same breed. That's a great question. So let me give you an example. Bull A has a plus 40 EPD for weaning weight. So its children, its offspring, are generally 40 pounds heavier than the breed average when they're weaned. And Bull B has a plus 20 for weaning weight. So both of them produce offspring that are heavier than the population average, but Bull A's offspring are 20 pounds heavier on average. Remember, this is a broad average. They're not going to all be 20 pounds heavier. The environment plays some role in this as well. So, you're not going to be able to 100% predict this, but it's a way to gain information when you're making selections about the bulls. Now again, none of this at this point is genetic in terms of I don't have a genetic test. There's a lot of genetics underlying this, but I haven't looked at any DNA information to inform my EPDs. Let me give you one more example before I take us to break. Let's look at marbling. So the USDA grades beef by its amount of marbling, and there's a numerical scale that they use. So if you have very little, whoops, back up, wrong button. If you have very little marbling, then it's called slight, and you have a very low marbling score. On the other hand, if you have a lot of marbling, you have a high score, and you get USDA prime. Now, you and I don't find USDA prime at the grocery store because prime is bought by all the top-end steak restaurants. You and I generally find choice and select in the grocery store. And choice costs more than select. So if I am going to sell my cattle, if I'm raising beef cattle, I want beef cattle that have a fairly high amount of marbling. Ideally, I'd love to have my beef cattle look like this, but there are other constraints against having too much marbling. But anyway, so let's say that I've got Bull C that has an EPD of minus 0.2 units, so it is less than the population average in marbling, and I have Bull D that has plus 0.2. So on average, if I'm trying to choose between which of these two bulls I'm going to use in growing my population, there's a 0.4 difference in marbling. And 0.4 might make the difference that moves me out of select and into choice. Or it might make the difference within choice that moves me into certified Angus beef. Because I could have an Angus beef that doesn't have enough marbling to be certified, and I'm going to get a higher price per head if it has enough marbling. So depending on which bull I choose, I may make a pretty significant difference in my pocketbook and how much money I'm going to get from this. Make sense? All right. So here are 4 bulls. I promise I'm going to give you a break in just a second. Here are 4 bulls, and here are some of their EPDs. Birth weight, weaning weight, yearling weight, and milk amount. Now this is the milk amount of the bull's daughters. A lot of the bull genetics, a lot of the paternal genetics controls milking. Traditionally, I will have to wait until this bull has given birth to multiple daughters, and those daughters have then reached maturity and given birth so that they are producing milk so that I know what I'm dealing with back for this bull. That can be 6 years and about $25,000 to $50,000 in animal costs before I can get this kind of information. But historically, that's the way we've done this. So if you look at these bulls, they've got different EPDs. Some of them, so this bull produces smaller than average calves. This bull produces much larger than average calves. If we look at their weight at a year, this same bull that produces the larger than average calves also is producing pretty big yearlings, and again are smaller than average, pretty small yearlings. But if I look at milk quality, the bull that makes the biggest calves, the moms don't give a whole lot of milk. They're not good milkers. And here, small calves, good milk. C, small, average size calves, even better milk. So if I'm trying to make decisions about what I want my herd to look like, if I am only interested in bringing the largest animal to market and getting the most profit I can, and I don't mean, that sounds like I'm saying that in a bad way, I don't mean that way, but if I'm not worried about keeping some of my cows because I'm going to then impregnate them to grow my herd, I'm going to sell all my herd, then I'm going with bull A as the dad, because I'm getting big yearlings, and I don't care if the milk quality's not so good. But if I'm going to hold back some of the cows because they're going to become the moms of my next generation, that is not a good option for me. Maybe what I want is I want smaller calves and I want better milk quality. Or maybe the cows that I'm going to keep, that I'm going to impregnate, maybe they've got big bones and they can have big babies. So I'm going to go with average sized calves and really good milk quality. So a farmer has to make these decisions based on what they want their herd to look like, using these EPDs as a guide. Okay, when we come back, we're going to talk about genetic markers. We're going to finally get into the molecular markers. So, 10-minute break, and then we'll come back and we'll get started again. So in 2009, the cattle genome was sequenced. So the collection of genes that make up the cattle genome. It was sequenced in 2009. Cattle share about 70% of their genetic information with humans. We're both mammals, so that's not terribly surprising. They have about 22,000 genes, not far from the same number, close to in the same ballpark as the number of genes that humans have, also not surprising. And there's a test now that you can offer you can take a sample of, pull out a cow's tail hair, or you can take a punch from their ear, take some blood, and you can do a genetic test. And essentially, they're looking at 50,000 sites in the DNA that vary, where there are alleles. Most of these are single nucleotide polymorphisms. That means there's a one-letter difference. One cow might have an A at this spot, one cow might have a C at this spot. And many of these 50,000 spots we've been able to tie back to some sort of genetic, some sort of physical trait. So for about $17 a head, you can have the 50,000, the 50K marker test done. The test is done on a glass slide. This is called a microarray. It's the same kind of thing that we actually do with human research. Dr. Devin Absher, one of the scientists here at the Institute, does microarray analysis, not on cows, but on humans, but it's the same kind of approach. And then there are a number of traits that have been identified. Now, some of this is proprietary, so you don't necessarily get all the information back when you ask the companies about it. But for example, I showed you the Texas Longhorn. There are a lot of ranchers that don't want horns on their cows. They cause damage to the other cows. The cows get caught by their horns, so they actually dehorn the cows. That is an extra expense, and it's stressful on the cow, and there can be things associated with that. So there is a genetic trait, there's a DNA change that actually leads to cattle that have no horns. It's called the polled marker. And so you can test to see if your steer and if your cow have those variants, and you can determine the likelihood that you would end up with offspring that don't have any horns. The same is true of coat color. Black coat color versus red coat color. Different patterns of coat color. Susceptibility to infections, how long the animal is likely to be fertile, and there are even some genetic changes that they're associating now with the degree of marbling or the degree of tenderness. So now you can actually begin to take, I should have done this first. As an aside, this is your trivia for tonight, plus the Miss Piggy piece. That was a free one. Anybody know the connection between cattle and HudsonAlpha? You're first in line for cookies next week if you know it.
They're both full of bull.
Oh! They're both full of bull. You are last in line for the cookies next week. Fair enough. Okay, here's the story. Jim Hudson was at UAH. He was in graduate school at UAH. He'd gone back to get a graduate degree as an adult. And he started a side business looking for genetic markers that were associated with muscle mass in cows. But in order to do this kind of test, you have to have starter fragments called primers of DNA, and those primers are artificially made. And at that time, there was one company making these fragments in California, and there was a really long, like a month-long backlog. Not because it was hard to make them, but because they were the only company making them. So Jim Hudson got a loan and spent $25,000 in 1987 and bought one of these machines that made the artificial primers. And right about that same time, this thing called FedEx appeared on the scene. And Jim promised that anybody that ordered these primers from him instead of the competitor, that he would get them to them within like 3 days. And that was the start of research genetics, which was the start of Biotech in North Alabama. And so when Jim and Lonnie McMillan started HudsonAlpha, there had been this long history in Huntsville of biotech, which really all started because Jim wanted to study muscle mass in cows. So, there's the connection, a nice side note. And my appreciations to Kathleen Shaw, who's our communications, who manages our communications group, because she brought that tidbit of information to me, and I'm very appreciative of that.
I didn't think Lonnie would be interested in muscle mass.
Lonnie was not involved in muscle mass in cows. Yeah, Lonnie's important contribution to HudsonAlpha and Lonnie's involvement in HudsonAlpha came after muscle mass in cows, definitely. So what we now can do with these EPDs is we can now take that genetic information from that set of 50,000 SNPs, that 50,000 marker, and add that in. So we can now take a genetic test the first day that a calf is born and look at this kind of information. So we can incorporate genomic information into EPDs, and so now they're called, if a farmer chooses to use genetic information, they're called genomically enhanced estimated progeny differences. And it adds the confidence equivalent to a bull having 8 to 20 offspring right off the bat. So you now know that much more information just from looking at the genetic markers. So it changes part of the way that it changes the accuracy or the confidence if you're making selections about who is going to set up the next generation of your herd. And so you get this accuracy much earlier in an animal's life. Only about 5 to 7% of cattle are currently using this. It's still very much in the early adopter technology, but it is beginning to be used more and more because some buyers are now offering premiums on the cattle that have this kind of information. And if it costs $17 to $20 a head, and you actually have a $50 to $100 increase, that's a worthwhile investment. It's especially useful if you're trying to figure out how to build your herd. Now, we're going to talk only very briefly about assisted reproduction techniques here. But the concept is that in the cattle industry, and maybe in other industries as well, but I know the cattle industry, you can actually super ovulate your female cow. So you give her, you inject her with hormones that cause her ovaries to release large numbers of eggs. So I can get lots of eggs. And then I can do one of two things. I can retrieve those eggs, and so now I have the eggs in a Petri dish and I can fertilize them with a bull's sperm, or I can go through what's called artificial insemination, and you get bull semen in like straws, in tubes, and you actually turkey baster inject it in. There's a little loss for words there. Now I'm probably going to be really embarrassed, but anyway. And so you artificially breed the cow. No, the bull does not have to be there. You've got everything that you need. So you can do it that way, and then you actually can flush out, 6 days later, flush out the fertilized embryos that are like 8 to 10 celled embryos. Or if you gathered the eggs to begin with, you can fertilize them in a Petri dish with the bull sperm. Either way, you've got a whole bunch of embryos from the cow and the bull that you want because you've looked at their genetic information. And then you return them, you implant them in a recipient, in a surrogate cow, which is a female cow that's been given hormones to behave as if she's about to be pregnant. So I can take a bull and a cow, and I can get lots of embryos, and I can put each one of them in a different recipient, in a different surrogate. So I now have a whole bunch of siblings born at the same time. This dramatically increases how quickly you turn over or how quickly you build your herd. And I can now use genomic information, so I'm not just using those previous EPDs, I'm now using genomically enhanced EPDs to make the selection about who my bull and who my cow need to be. So one of the things that this does is that the generation time shrinks from 4.3 to 2.2 years. So I almost cut it in half with how quickly I can now bring the next generation to bear. So this is a game changer. This is where genomics makes a big, big difference in cattle breeding and in shaping the EPD information. Let's talk about chickens. 20 minutes to cover chickens. Woo! All right, so poultry were also domesticated. Poultry were domesticated primarily from the red jungle fowl, although we know that there's a little bit of gray jungle fowl as well, because chicken has yellow skin. That came from the gray jungle fowl. The red jungle fowl does not have yellow skin. So somewhere in this domestication event, we got a little gray jungle fowl in there as well. And this domestication happened multiple times all around the world. So chickens were domesticated simultaneously in lots of different places. But until the 20th century, they played relatively small roles in the American or worldwide diet or in the economy. They were bred, they were domesticated originally because they were fighters, not because they laid eggs or because we ate them. So, and then in the early 20th century, you had them on your farm, but it wasn't, you didn't get a whole lot from them. You got a few eggs, and when they quit laying eggs, you suddenly, you had chicken soup. But it was not a major driver of our diet or of our economy until really starting in the 30s and 40s and definitely in the 50s, when we began to commercially grow and raise chicken. Now, just like we have beef cattle and dairy cattle, we have broilers, which are raised for their meat, and we have layers, which are raised because they produce eggs. Now, both do this, you can eat a layer and you can get eggs from a broiler, but if you're trying to breed them, you either have to put your energy into making really good eggs for a lot of your life or growing lots of muscle mass. It's very difficult to get a chicken that does both. So they split the breeds, and this one set of breeds became focused on meat, and one set of breeds became focused on laying eggs. And here are some of the commercial types. There are about 4 or 5 companies around the world that provide most of the chickens for either layers or for broilers. One of the largest is here in Huntsville, Aviagen. They're headquartered here in Huntsville. And there's some really incredible science that goes on both here and also in Scotland, where much of their R&D work takes place. They're one of the main players in the poultry industry. And very, very nice people. They gave me a lot of background to help me work through this, because I am not a chicken farmer. I do not know this content naturally. And then in addition to the commercial varieties, there are all kinds of exotic and heritage breeds that you might grow in your backyard. At least I think in parts of Huntsville you can have chickens in your backyard. Look at this. Look at this one, golden Polish. Those are the feathers on top of its head. It is not wearing a hat. That is what it looks like. The French black copper Marin is a layer, and you will spend several $100s for a half a dozen of its beautifully ruby-colored eggs. Yeah, several $100s for half a dozen. Yeah. So lots of heritage varieties as well. Now, these don't work well growing commercially. They don't give enough eggs. They don't lay over a long enough period of time. They don't do well under commercial growing conditions. But they're kept as, as heritage varieties in people's farms and in their backyard. The breeding platform for poultry is that there is a set of breeding stock, purebred breeding stock, that have lots of different traits. And the poultry breeders decide, okay, well, this one produces, let's say that we're going to focus on eggs. This one produces great-sized eggs, but doesn't produce as many eggs as I would like. This one produces lots and lots of eggs over a 5 or 6 year window, but the eggs are small. Can I breed them and crossbreed and back cross them so that I get bigger eggs that are produced through a longer number of years? And so that process of going from the breeding stock to the great-grandparents to the grandparents to the parents, and then all the way to the broilers or layers that are sold to the commercial chicken farms, that's about a 4-year window to move from, hey, what if I try this set, to this is now what's being made, this is what's now growing in the farm, the chicken farm that's down the road. About a 4-year window. These next 2 slides are amazing to me. Chickens are genetically, there's so much variation in the chicken that we've been able to take advantage of for growth. This is the time that it takes for a chicken to get to market weight. And this is the weight, the market weight. So you can see in 1925, market weight was about 2 pounds, and it took over 100 days to get to 2 pounds. Today, market weight is about 6 pounds, and it takes about 40-something days to get to market weight. Oh, I'm so glad you asked. Those growth curves have nothing to do with hormones. You are absolutely correct. Chicken are not fed hormones. That is a myth. Chicken, beef, every livestock is fed low levels of antibiotics. So let me come back to that. But chicken are not fed hormones. Chicken are not fed steroids. If you were going to give chicken growth hormones, you actually would have to inject it, because the kind that you would eat are broken down by your digestive system. So you would have to inject it. And chickens, you would have to inject chickens multiple times a day, and the average chicken farm has 20,000 chickens. So when you see on a piece of chicken, when you see on the package it says grown without hormones, so is every other chicken on the rack. But because there is this urban legend that chickens are grown with hormones, some of the chicken companies felt they needed to put language on there saying theirs are not grown with hormones. So I want you to hold to that, Mary, because the next set of images I'm going to show you, you're going to look at that and go, how is this possible? They are not grown with hormones. Now, they are grown with low levels of antibiotics, and we talked about this last year when we did the microbiome. They're given sub-therapeutic, which is lower than the amount you would take if you had an infection, levels of antibiotics. And that seems to improve their growth. That seems to increase their growth. That's consistent across most of the different meat industries. And that's a separate issue that we can take up later. But what I'm showing you, the increase in market weight, the decrease in time to market weight, and this feed conversion, which is how much food does it take to get a pound of weight on the chicken. We now have about 2 pounds of food equals 1 pound of weight for the chicken. In beef, it's 7 pounds of food for 1 pound of weight, and in pork, it's 4 pounds for 1. This is all because we have selected grandparent and parent strains that grow well. And because we've got multiple generations, you can do multiple generations of chickens in a year, so you can put some pretty serious selective pressure to get the strains, the breeds that you want. This is it's amazing. Now, let me show you this next picture. This is looking at breeder stock, so the kind of chicken that you would send to market from 1957 using the strain that was available then, 1978, which was a different strain, and 2005, which is the same strain that that currently is used in the market today. Now, I don't know if you can see these numbers. At birth, the 1957 broiler weighed 34 grams. Today it weighs 44 at birth. At 56 days, in 1957, you got 905 grams on average. 4,202 grams. Again, these chicken are not fed hormones. These chicken are not fed steroids. And these chicken have grown 400% larger. Let me say that differently. The market weight of these chickens is 400% larger than the 1957 breed on half as much food, because they have genetically selected those chickens whose cells maximize the energy out of the nutrients and grow rapidly. This is domestication and genetics, and they're not doing genetic testing. There's not DNA testing. This was based solely on, I am physically going to see the chickens that grow fast, and I'm going to use that in my breeding scheme. This was not, let's do a DNA test. This is the whole process of domestication.
Got anything to do, they don't get to walk around, they're in cages now, or?
So how much energy would they normally expend if they were free? They certainly are moving around less. So I don't know how much of that are they not expending it? Chicken don't normally flap their wings, not a whole lot, which would work the breast muscles, which is where most of this growth is. So I'm not sure how much of that's because they're just not burning the calories off versus they're just much more efficient at packing the meat on.
Broilers aren't in cages.
What?
Broilers aren't.
Broilers are not in cages, they're in farmhouses. Very good point, Francis, thank you. They're in farmhouses, so they can move from one end of the farmhouse to the next. So they do have that option. And this is the side view of the birds as well. Now, let me tell you, this comes at a cost, because this kind of rapid growth means that we now suddenly are dealing in the chicken population with the same kind of issues that we're dealing with as humans. As our population gets heavier, we see increases in weight-related traits. So for chicken, in the 90s, as this really took off, the early 90s, there was an increased incidence of cardiovascular issues where they couldn't oxygenate their blood to high enough levels, and they began getting fluid buildup because there was so much more weight that they were trying to pump blood through. In the same way, as they put more weight on, there began to be a greater incidence of leg deformities. So the growth plate in the tibia began to, it would separate, the legs wouldn't form correctly, they couldn't support their weight. So they then developed X-ray machines that looked at the tibia and looked for these weaknesses. And they did this at the breeding level, at the breeding stock or the great-grandparent level, and they were then able to only select the chickens that could oxygenate well, that had stronger hearts, that didn't build up fluid, and that didn't have leg weakness problems, and they put that into their breeding scheme. So they were able to selectively breed for those chickens that could handle the increased weight best. And you can see here, so this is showing you the incidence of tibial dyschondroplasia, which is the leg issue, the leg weakness, the leg deformities. And you can see that in 1990, when they first introduced the X-ray machine for chicken, that the incidence of this hovered between 25 and 30 to 35%. So a really large portion of the chickens that they were testing had these leg problems. But then pretty quickly, the incidence dropped as they began to only breed up in that great-grandparent level, the chickens that were strong. They pulled out from the breeding stock the chickens that had the leg problems. And remember, it's a 4-year window to move from that early stock all the way down to the broilers. So the incidence dropped. And then in 2008, another second-generation machine was developed that actually identified it even more accurately. So chickens that with the first-generation machine, you wouldn't have seen it. The second-generation machine detected it, which is why the rate went back up and then is coming back down. So even though, let me back up. There are unintended consequences to every selection that we make. Sometimes we select a specific plant because of the way the fruit looks, and we select out resistance to pests, or we select out drought tolerance. That's an unintended consequence. There are unintended consequences when we select for growth, or when we select for long fertility. So we always have to deal with that. We have an unintended consequence of being humans that are designed to extract as much energy out of food as we possibly can, because evolutionarily speaking, we didn't know where the next meal was coming from. We don't have that problem today. So our bodies continue to extract as much nutrients out, and we're getting less exercise, and we're now dealing with those consequences. So the point that I want to make is that yes, there are things that we have to deal with when we breed for a certain trait. If you know dogs, you know that there are lots of dogs because of inbreeding that have heart problems, that have leg problems, that have hearing problems, that have spine problems. That's exactly what we've done. We've brought that on. But if you also at the same time are selecting against some of those issues and you're picking the fittest breeders that you can get, you are increasing the overall health of the entire flock, ultimately, of the entire commercial industry. So genetically, the chicken genome was sequenced in 2004, has about 17,000 genes. It's only about 7% similar to us. Birds and humans share a much, much more distant common ancestor than cows and humans, both being mammals. So about 17,000 genes. The paper was published in Nature, and there was news and views written about it, about what the chicken genome would bring. And the authors of that news and views are Jeremy Schmutz and Jane Grimwood, who head the HudsonAlpha Genome Sequencing Center. This was written right at the time that they were finishing the human genome and getting ready to transition into agriculturally important crops. So you hear me talk about Jane and Jeremy all the time in the last 3 weeks, but I just wanted to bring that point up. Now, the process of identifying genetic markers associated with traits in chickens is moving much more slowly than what I showed you in cattle. We don't have as much information yet. So if you go back to the analogy I showed you the very first week when I talked about drawing the boundaries of maps and the early boundaries of the United States, we kind of knew the coastline, but we didn't know anything about the interior. We didn't know where the major rivers or the mountain ranges were, and it took exploring that in depth to find those individual land features. That's where we are with the chicken genome. There's a whole lot that we don't yet know about the chicken genome, especially how it might be useful in adding information for a breeding scheme. But I want to give you 2 genetic examples before we wrap up. The first one's a gene called TSHR. TSHR stands for thyroid stimulating hormone receptor. So let's break that down a second. That last word, receptor, that means that it makes a protein. Genes provide instructions. Most of the time, those instructions are for how to make a protein. This is a protein that sits on the surface of a cell. Think of it as a baseball glove. And this baseball glove is designed to catch a very specific shaped ball. That ball is thyroid stimulating hormone that's produced by other parts of the body that travel through the bloodstream. So when the other parts of the body produce thyroid stimulating hormone, which one of the things that hormone does is stimulate the thyroid, this receptor, this protein, this ball glove, sits on the outside of the cell, and if this hormone is present, it catches it, and it signals to the cell that, hey, this hormone is out here, we need to do something, we need to behave a certain way, we need to turn on certain genes. This whole process, hey, we need to turn on certain genes, coordinates the reproduction window of the chicken. So when is the chicken able to reproduce? When is it cycling and able to breed? And it's actually correlated, the production of this hormone is correlated to the amount of daylight, which means it's tied to the seasons of the year. So ancestrally, in the ancestor, the wild bird, you only bred during certain times of the year. You didn't want to be breeding and laying eggs in the middle of winter. So there were only certain times of the year when you could go into breeding. What's interesting is that domestic chickens, every domestic chicken has a mutation in this gene. So it's inactive, which means you can breed and lay eggs year-round. That's really advantageous if you are breeding chickens for food or for lots of eggs, that they don't only breed a certain window of the year. That change, that mutation, isn't present in the wild version, the red jungle fowl. So that mutation somehow developed or was selected for multiple times, because I told you chickens were domesticated in lots of different times around the world. And you can imagine, if you are trying to domesticate chickens, you're going to definitely pay attention to that chicken that is producing eggs or producing chicks year-round. And you're going to select that to go into the next generation of your breeding scheme. So the mutation occurred randomly. We didn't force the mutation to occur. And then the breeder took advantage, or the farmer took advantage and selected that and used that in the next generation. Last gene, TBC1D1. This is a really intriguing story. This gene is involved in the regulation of moving glucose around. So it's really important to getting glucose in and out of your cells and from one cell to another. In humans, we discovered, the first time we learned about this gene was in humans because we discovered it was associated with body mass index, with BMI, and with waist circumference. And there were certain changes, certain alleles, variants in this gene in humans that were associated with larger waist and with higher BMI. Not so good if you're a human, but great if you're producing livestock to come to the table. So they then went back and looked at this gene in pigs and looked at if there were any changes in this gene in certain breeds of pigs. And it was found to be associated with fatness in pigs. So pigs that were larger, that had higher levels of fat, had mutations in this gene. So now we found this in humans. We've seen it in another organism, in pig. In broiler chickens, they looked at lots of different varieties of broiler chickens, and the broiler chickens that had changes, that had alleles, variants in this gene, had higher body weights, carcass and eviscerated weight, and the eviscerated weight's important because that's what the consumer is buying, not the internal organs, and higher leg muscle weight. So while this marker is not yet something that we're going to use in the breeding scheme, it's got to be confirmed in lots of other studies, finding this kind of genetic information may help shape how we pick strains in the future. All right, so we've covered a lot of livestock, a lot of this. I would hope that you've seen some of the same kind of stories we talked about in crops last week, the same kind of selection process, and how knowing the genetic information helps shape and inform that process, that you'll see those same things showing up in livestock conversations this week. Next week, we're going to talk about genetically modified foods. We're going to spend the whole time on genetically modified foods. We're going to talk about what they are, how they're made. We're going to talk about the things that people are worried about with them. We're going to talk about the potential pros, the potential cons. We're going to dig through the hype, and there is a lot of hype. A lot of hype. And then we're going to talk about some emerging technologies that may or may not be considered as genetic modification, and I'm going to ask some questions of you as to whether you think they are genetically modified or not. So we're going to cover a lot of ground next week. I look forward to seeing you next Tuesday for our final Biotech 201. Have a great week and stay warm, everybody. Take care.